Mitosis and Meiosis by Meg and Amanda. Before mitosis, interphase occurs. During interphase, the cell grows and copies its chromosomes to prepare for cell division. This consists of the G1 phase, where the cell grows, the S phase, where the cell copies its chromosomes and replicates the cell's DNA, and the G2 phase, where the cell continues to grow and prepare for cell division. Now mitosis is ready to begin.
As mitosis begins in somatic cells, prophase occurs. During prophase, chromatin fibers become tightly coiled into chromosomes, each consisting of two identical sister chromatids. The meiotic spindle forms composed of centrosomes and microtubules.
Next, metaphase occurs. As prometaphase occurs, the nuclear envelope fragments and microtubules invade the nuclear area. The chromosomes become more condensed, and each sister chromatid has a kinetochore, a specialized protein located at the centromere that jerks chromosomes back and forth.
It is now time for metaphase to begin. It is the longest stage of mitosis. The centrosomes are now at the opposite poles of the cell, and the chromosomes line up on the metaphase plate.
This phase was about 20 minutes long. Now it's time for anaphase, the shortest stage of mitosis. This begins when the cohesion proteins are cleaved. This allows the two sister chromatids to part. Suddenly, each chromatid has become a chromosome. The two free chromosomes move toward opposite ends of the cell. By the end of anaphase, the two ends of the cell have equivalent, sorry, I can't pronounce anything, and complete collections of chromosomes.
In telophase, two daughter nuclei form the cell along with other portions of the parent cell's endomembrane system. The chromosomes become less condensed and mitosis is complete. Next, cytokinesis, which involves the division of cytoplasm, occurs. In animal cells, a cleavage furrow pinches the cell into two, and the cytoplasm divides. In plant cells, a cell plate forms instead of a cleavage furrow. This marks the end of mitosis. Now it's time to talk about meiosis. In gamete cells, cell division occurs with two parent cells. Fusion of two haploid gametes, a sperm and an egg, results in a diploid zygote, which divides by meiosis, where a single homologous pair of chromosomes are replicated and then sorted into four daughter cells.
Prophase 1 is the first phase of meiosis, where chromosomes begin to condense and align gene by gene. Crossing over occurs here, while homologs are in synapsis and the chromosomes in each pair move slightly apart. Centrosomes move, the meiotic spindles forms, and nucleus envelopes break down. In late prophase 1, microtubules attach to kinetochores and the homologous pairs then move toward the metaphase plate.
In metaphase 1, pairs of homologous chromosomes are aligned on the metaphase plate, with one chromosome in each pair facing each pole.
Next is anaphase 1, where homologs separate and move toward opposite poles.
Next, in telophase 1, each half of the cell has a complete set of replicated chromosomes. And cytokinesis occurs during telophase 1, forming two haploid daughter cells, each with its own cytokinesis.
Next we have prophase 2, where a spindle apparatus forms and chromosomes move toward the metaphase 2 plates.
In metaphase 2, the chromosomes are positioned on the metaphase plate. The kinetochores are attached to microtubules extending from the opposite poles.
Next we have anaphase 2, where the kinetochores holding the sister chromatids together break down and the chromatids separate and move toward opposite poles.
Next is telophase 2 and cytokinesis. The nuclei forms, chromosomes decondense, and cytokinesis, the division of cytoplasm, occurs. Four daughter cells are now formed, each with a haploid set of chromosomes. This concludes meiosis.